Hey guys, so since today is pretty much go at your own pace, I wanted to record this video now to go over the cell membrane and cell transport notes. Once I am done, I'm also going to spend the time to look over the assignment for those of you who don't really get how to complete the Cornell notes or want some guidance in what you need to have at each moment, so you can stay for that. If you want to, while you're watching the video, you do have the option to kind of work on those notes while you're doing it, but you don't have to. You can kind of sit back, relax, and absorb the information and then put that information to work when you are completing your Cornell notes. Okay. So the title is the cell plasma membrane and cell transport. I have it as cell transport for short. There are two essential questions, which I just put out as essential questions on this presentation instead of just calling them EQs. The first essential question is, what are some of the main functions of the cell membrane? I'm going to give you several. You're just going to give me a few of them. And the second essential question includes what is the difference between passive transport and active transport? And I do want you to include one or two examples for each one of those. Okay, so when you're looking at cell boundaries, there's two main types of cell boundaries that we have learned about so far. We have a cell membrane, which every single type of cell contains, and it allows things to pass through. It's a very flexible barrier. You also have a cell wall, which is only present in plant cells of eukaryotes, and a cell wall is present in all prokaryotes. A cell wall is a strong supportive layer, so it provides more support and protection for those types of cells. So for our cell membrane is made up of what is known as a phospholipid. In fact, the cell membrane is called a phospholipid bilayer because it's made of two lipids that are put close together. Now, a phospholipid is made up of a hydrophilic head, which is the ball portion of a phospholipid, and a hydrophilic actually stands for water loving. The hydrophilic head enjoys water, so it can be connected to a water environment inside of the cell and on the outside of the cell. The phospholipid is also made up of a fatty acid. Remember, fatty acids are monomers for lipids, and when you think of lipids, you kind of think of oils. Water and oil never mix, so the fatty acid tail is known as hydrophobic, meaning that it fears water. It doesn't really fear water, it just doesn't like water, since once again, lipids and water do not mix. If you poured oil and water together, they're just going to separate out from one another. And these phospholipids come together to make the membrane, and it allows the membrane to be selective, meaning that it allows it to pick and choose what material it is able to let in and what material it is able to push out without allowing everything to come in. So just to get a closer look of what that hydrophobic head looks like and the hydrophilic tail, now the membrane, which we're going to look at, is actually made up of two phospholipids. So you have a phospholipid bilayer. On the outside of the cell, you have the hydrophilic head on the outside. You have your tail on the inside that is connected to the other tail of the other layer of phospholipids that is a part of the cell membrane. And then you had your hydrophilic head on the outside. So the non-polar, the hydrophobic part of the membrane is on the inside, and the hydrophilic, the water loving part of the cell membrane, is on the outside of the cell and on the very inside of the cell. So this is just a close up of what the cell membrane looks like. You see your lipid bilayers here, with the round balls being the heads, and on the inside, that's where you have your hydrophobic tails. These purple structures that you see here are proteins. Proteins allow for those larger molecules to pass through. Think of glucose or amino acids that need to get into the cell in order for the cell to build the proteins that it needs. And you can see that these phospholipids are really close together. Small molecules are able to easily transport through, but those larger molecules are going to need those proteins, which we're about to discuss. So there are several functions of the cell membrane. Cell membrane does act as a protective layer. It definitely helps regulate and transport material into and out of the cell. It allows for cell recognition with other cells or different tissues that may need it. It helps provide anchoring sites for filaments and cytoskeleton. It provides a binding site for enzymes. You have interlocking surfaces that help bind cells together. So how these cells are able to interlock in order to form things like tissues, which is the next level of organization. And also it contains cytoplasm, which is on the inside of the cell, which is that liquid part of the cell that the organelles are floating inside of. So our cell membranes are known as semi-permeable. A semi-permeable membranes allows for things like small molecules that are able to transport through. They're able to fit through the different spaces within that phospholipid bilayer, and they're able to flow through and out of easily. So examples of these small molecules, oxygen is definitely one, carbon dioxide, and also water, or examples of these small molecules that are able to go into and out of the cell. So ions, hydrophilic molecules that are larger than water, and large molecules like proteins cannot move in and out of the membrane by themselves. So they're going to need a larger structure to help them move through, like the proteins that we saw on the picture diagram of the membrane. So the main goal of the cell is to maintain homeostasis. So pay attention to this word. It is definitely going to come up several times throughout the year, but we're going to be focusing on homeostasis within the cell. Homeostasis is the cell's need to keep everything balanced. It wants those conditions to be maintained or to stay in a state of equilibrium where there's not much change occurring. The main thing that maintains homeostasis within the cell is actually that plasma cell membrane. So they're able to dictate we need more oxygen within the cell. Let's get rid of that carbon dioxide. We need more water within the cell. We need more salts. We need more amino acids to maintain the homeostasis. And the plasma membrane is able to dictate, let more of those things in or take some of those things out. So there are two different types of transport, passive transport and active transport. Starting off with passive transport. Passive transport is a type of transport that requires no energy. So no energy is used for these materials to enter or exit the cell. It happens naturally. So typically molecules like to move from an area of high concentration to an area of low concentration. So we're looking at concentration, like how much a material is dissolved within that particular solute. When you think of concentration or the concentration gradient, I like to picture a hill and think that you have bowling balls or something round that can easily roll down that hill. You can definitely have that ball that you put on top of the hill roll down the hill when it's in a high area to that low area naturally without inputting any type of energy within it. So one form of this passive transport is diffusion. So diffusion is a passive process. It means that no energy is used, and it allows molecules to move as they would naturally. So their kinetic energy, the way they like to go. And molecules like to move down a concentration gradient, meaning if they're in an area of a higher concentration, they want to move away where there's an area of lower concentration. Kind of like if you were at a concert or you were in a room and everybody was tightly packed in this one area, where you're going to move where there's more comfort and more space in another part of the room where nobody else is. Molecules like to do that as well, and it does it naturally through the process of diffusion. So when you're looking at diffusion of liquids, if you guys ever took water and placed a couple drops of food coloring within it and watched that food coloring slowly start to spread out, that is an example of diffusion. So as those molecules are spreading out, once again, they're moving from the high concentration, which is the initial drop of food coloring that you put within the water, and it moves to an area of low concentration, which is everywhere else within that droplet of water. And they continue to spread and move apart from one another until there's equal concentration of that solute throughout the entire glass. So you can see that here with the high concentration of that dye that was placed in there, it starts to diffuse or spread out, moving down that concentration gradient from a high concentration to a low concentration. And then it stops when everything is equal, when everything is easily spread. So when you see in this picture up here, everything has that kind of pink coloring within it, then that's when it's going to be even and the diffusion process is going to start. You can also have simple diffusion within gases. So an example of diffusion in a gas, if you ever took a bottle of perfume and opened it up and then set it down and then walked to another part of the room, eventually with time, you will start to smell that perfume on the other side of the room. What happens is the molecules within that bottle of perfume turn into a gas. And like all molecules, they like to move from an area of a high concentration right above that perfume to an area of low concentration, which is everywhere else in the room. So eventually, everybody in my classroom will be able to smell that diffusion with that perfume, which is an example of diffusion. So simple diffusion that occurs within the cell without needing anything larger, things like oxygen or water, which diffuse usually into the cell, and carbon dioxide, which is a waste byproduct when we go over photosynthesis and respiration. Carbon dioxide is a product of respiration, and that needs to leave the cell and diffuse back into our blood. Now, another type of diffusion is known as osmosis. Now, osmosis is a science term that a lot of people know, but people constantly use this term incorrectly. So, an example, I'm hanging out with my friends. This happened a few months ago, and they talked about, man, I wish I would go back to school if I could just absorb the books, like the information from a book by touching it, and just absorb all the knowledge from the book through the process of osmosis. And I'm there and I'm like, goodness, I'm going to have to be the science teacher hanging out with my friends. And I'm like, well, actually, that doesn't happen that way. Here's the reason why. Osmosis is the diffusion of water. The only thing that can go through the process of osmosis is water. So that is diffusion of water through a semi-permeable membrane. Water liking to move from a high concentration to a low concentration, and this happens naturally. Only water can be involved with osmosis. If you're talking about anything else, once again, unfortunately, we cannot touch a book and absorb its knowledge, but let's say that was possible, that would just be an example of diffusion. Anything else that naturally moves from an area of high concentration to an area of low concentration, that is diffusion. It happens naturally. But if you have the transport of materials across a semi-permeable membrane and it's just water, it is osmosis. Make sure you understand the difference between those two. Osmosis is just the diffusion of water. If it involves any other type of molecule, it is not osmosis, it is just diffusion. So let me step off my soapbox. And if you guys walk out of this class knowing at least that osmosis is a diffusion of water, and you know that it's just a type of diffusion, but only water can go through osmosis, then I've done my job. So the diffusion of water, once again, goes from an area of high concentration to low concentration. So if you kind of think of how much solute is actually dissolved within that water, on this side of the cell, it has a high concentration of that solute, the chemical that is dissolved within the water. On the outside, you have a higher concentration of water molecules, but a low concentration of that solute. So water is going to naturally move to the high concentration of that solute, because it's going to go from a high concentration of water to a lower concentration of water. So facilitated diffusion, this is for the molecules and compounds that are too large to easily move through that semi-permeable membrane, so they need a little bit help. Facilitated diffusion still does not use any energy, so it is still an example of passive transport. So it uses what is known as transport proteins, those purple structures that you saw on that, that diagram that I showed you earlier on. It uses these transport proteins, which are much bigger. They allow for those, they are made for those particular type of macromolecules that they're looking for, and it's able to take that larger molecule and move it on through. So it uses no energy because this is something that happens naturally. Molecules naturally like to move from an area of high concentration to an area of low concentration. So since it's following that rule, no energy is used. An example of some of the chemicals that they're looking for is glucose, what is used to build carbohydrates, are used to be broken down in order for our mitochondria to create the power that our cell needs, or amino acids, which are the building blocks of proteins, and we have a bunch of ribosomes within our cells that need to build these proteins, so that's where it gets the amino acids from. These two compounds are brought into the cell through facilitated diffusion or through those proteins within our cell membrane that help bring them in. So an example of facilitated diffusion, once again, it goes from an area of high concentration to low concentration. We have a high amount of that material on the outside of the cell, and so it's going to naturally move through the protein and into the cell because it has a lower concentration on the inside of the cell. Now, the different types of solution. There are three different types of solution when you're comparing them. The first is isotonic solution. An isotonic solution is a solution of equal concentration to a cell. So if you have a cell, you place it in the solution, the concentration of that solute is equal inside the cell or into the cell. Things still flow in and out of the cell, but there's no major changes because there's no concentration gradient where things can move from high to low. When it's equal, things kind of stop. You have a hypotonic solution. A hypotonic solution is a solution of a lower concentration than a cell. So this means that it has a high water potential, meaning it has a low amount of dissolved solutes when you're comparing it to that of what is within the cell. And you have a hypertonic solution. Hyper is the exact opposite of hypo, where you have a solution of a higher concentration than the cell. So this has a low water potential. So whenever you're using these three terms, it's when you're able to compare concentrations of solutes. If you're not able to compare solutions, then you can't use any of these terms because you're not able to tell if it's iso, hypo, or hyper. You can only tell if you have another solution to compare it to. So what does that do to the cell? What does it mean? So if you took a cell and you placed it in an isotonic solution, so in this first picture that's in the upper left, you have a cell. Inside of the cell, it has a 10% salt concentration. Outside of the cell, the material that it is in, it also has a 10% salt concentration. Water goes back and forth between both directions, just kind of flowing easily when this happens. Now, if you took a cell that was and placed it in a hypotonic solution, meaning that it had less of that solute. So you had a cell that had 10% salt solution on the inside of the cell. You placed it in a solution that had a 10% salt solution, so it had a lower concentration. So during this time, water is going to move inside of the cell. The reason why, that water wants to move inside to dissolve the salt that is within the cell to make it equal to what it is outside of the cell. So this can actually have damaging effects to the cell if this would occur. And then you also have a hypertonic solution. So for this example at the bottom, you have a cell that has a 10% salt concentration. On the outside of the cell, it is within a 20% salt concentration. Once again, the cell is going to naturally allow water to exit and enter the cell. Water is going to move out of the cell because water wants to move from water is going to move to kind of dissolve that salt that is within there until it becomes equal, and this can lead to a disturbance of the cell as well. So how does that affect the cell if it's placed in a hypotonic solution versus a hypertonic solution? So in the middle, we have isotonic. Things are equal. Cells are nice and happy. Cells have the shape that they're supposed to have. If you took a cell and put it in a hypotonic solution, meaning that it had a lower concentration on the outside of the cell, water is going to rush into the cell, and it does it naturally. And this can lead to the cell being lysed. When the cells swell up, sometimes they swell up too much, especially when too much water actually enters the cell, and they lyse or they burst, so they can actually get too big. If you have a hypertonic solution, meaning you have a higher concentration of solute, then the water that is within the cell is going to move out of the cell, and this can lead to the cell looking shriveled since a lot of the water that was inside kind of exits, so kind of dries it up a little bit if you think of it that way. So we looked over passive transport where we did not use energy. Now we're looking at active transport where we will need energy. It requires energy or ATP. ATP is another form of the energy that our cells use in order to do the functions that it's meant to do. Active transport moves materials from a low concentration to a high concentration, which means that it's against the concentration gradient. So remember that heel analogy with the bowling balls on top that we use that naturally go down from high in the area to a low area? Well, now think of it this way. If you had to push the bowling balls up that hill, you are going to use a significant amount of energy than if you just allowed them to roll down a hill. So active transport has to use energy because it's going against the concentration gradient. So to give you an example, we have a low concentration on the outside of the cell, but we have a higher concentration on the inside of the cell. So energy is going to be used because naturally molecules like to move from high to low. So in order to do that, we have to use energy to pass it through these pumps that then pump the material that the cell will eventually need into the cell. So an example of active transport for this example, once again, when it's going against the gradient, is a sodium or potassium pump. It's essentially a pump that adds these elements into the cell that our body then uses to function, to use to create other things that we may need. So another form of active transport is endocytosis. Endocytosis involves the taking of material into the cell by means of enfolding. So it kind of goes up to the material that it wants. It then takes its cell membrane and circles the material, and then it forces that material into the cell. Another example of this is pinocytosis. Pinocytosis is very similar to endocytosis. The only difference is that pinocytosis only involves liquid. So they kind of coined it as cell drinking because it's only bringing in more liquids. Phagocytosis is very similar to endocytosis, but it's usually used for eating. So think of organisms like amoebas that are single cell organisms. And what happens is the same thing. The cell membrane engulfs that solid material that it wants and brings it into the cell. And then the cell materials within the cell is able to dissolve that piece of food that the amoeba may need. And then once it gets dissolved, then the other pieces within the amoeba are able to break that down and then use the energy that they got from the food and to use it for energy for the cell. So an example of this, if you guys ever seen the movie Monsters vs. Aliens, it's one of my favorite animated movies, but they have the blob there. And if you see the blob eating, he takes something in, he brings it into his body and you see it dissolve. That's an example of phagocytosis. Now, exocytosis is the opposite of those first three. Exo, meaning you want to push it out. So our cells, we have lysosomes that are constantly cleaning things up. They gather the materials that they need and they place it into a vacuole or a storage portion of that cell. That vacuole then moves up to the cell membrane, attach the cell to the cell membrane. The cell membrane is then able to open up, release all that material, and that process can continue again. So once again, we covered passive transport, which does not require any type of energy. It uses a concentration gradient where things naturally move from high to low concentration. That is a theme in science that you will hear several times while you are at high school. And then you have active transport, which moves things against the concentration gradient, meaning that it goes from a low concentration to a high concentration. And since it's doing against what it does naturally, it does require energy. Main difference between the two, one does not require energy, active transport does. So if you guys need assistance, you can stay for the other half of the video. What I'm doing now is opening up my Word document so we can look at the notes. So this is the Cornell notes. Things that you need to add. From the very first how to slide, you have your essential question. Please make sure you type those. I do want you to have my essential questions there because it will help you with your summary. When you're completing the notes, what you're going to add here, give me the two different types of cell boundary. Tell me some information about each one. Tell me for phospholipids, break down what phospholipids are or how they're made. You have 4 points that you need to pull from the slide to get that information. For the function of the cell membrane, I just want you to choose 3. I think I gave you about 6, so you're just going to type those functions in here. Semi-permeable membrane, I just want you to define it. For homeostasis, please define. And then I want you to answer the question, what maintains homeostasis within the cell? That was all answered on that same slide. Moving on to cell transport. For passive transport, tell me 2 facts about passive transport. And then all of these different types of passive transport and the different solutions, all I want you to do is define those. For active transport, give me some information about active transport. Essentially, use those bullet points that I gave you. And then for the different types of active transport, I just want you to define those words. So once you are done with your notes, you then need to create your own review questions. These come from you. So the best thing that you can do is read through your notes, think of a question that will help you understand the material, and then you're going to type that question over here. You do not need to include the answer because the answer comes directly from your notes. You can also think of a question for passive transport. So you can get your second question and do the same thing, read through your active transport notes, think of a question, and then you ask it over here. To get full credit on this assignment, you need at least 3 review questions. If you do not have 3, you're going to not earn credit. Now, if you put more than 3, you're good. I'm not going to take more away. So if you want to do 5 questions, go ahead and do that. But I'm looking for at least 3. Once you are done with your review questions, it's then time to work on the summary. Now, the best way to write the summary is to use the essential questions to help you. The summary needs to be a cohesive paragraph. I believe one of your questions is, give me a couple functions of the cell membrane. So you can talk about the cell membrane is made up of a phospholipid bilayer that allows for materials to enter and exit the cell. And then you can tell me a little bit more information about the cell membrane. And then the second essential question is asking you, what is the main difference between passive transport and active transport? You can give me some of that information. 3 things that I'm looking for in your summary. 1, it needs to be a cohesive paragraph. It needs to contain at least 3 to 5 sentences, and I'm looking for the answers to the essential questions. If you hit all those requirements and your summary is excellent, then you're good. Once you are done, make sure this document has saved, and then go back to the Canvas page, or you probably have it still open. Once it has said save, click submit assignment, and then you're done for the day. If you have any other questions, make sure you contact me, and good luck.